We learned the stages of mitosis, and now we have to learn the stages of meiosis. Now, here's the good news. If you've already felt comfortable with the stages of mitosis, then the stages of meiosis should sound very, very familiar. There's two rounds of cell division involved in meiosis, Meiosis 1 and Meiosis 2. They're preceded by interphase, and the interphase is exactly the same. You have G1, S, and G2. Okay, so all of those same events, duplicating the chromosomes, right, making sister chromatids for each chromosome, that happens before meiosis as well. Cell puts on mass, goes about its normal cellular functions, that happens before meiosis as well. So interphase is the same. The purpose of Meiosis 1 and Meiosis 2 is slightly different, though. The purpose of Meiosis 1 is to cut the number of chromosomes in half. So cells are haploid after Meiosis 1. Please write that down. Cells are haploid after Meiosis 1. Okay? Now, what we're going to do effectively is take the homologous pairs of chromosome number 1 from dad, chromosome number 1 from mom, and we're going to separate them to separate daughter cells. So each daughter cell will have half the number of the original parent cell, but it'll have one copy of each kind of chromosome. Then in Meiosis 2, we are going to break the sister chromatids apart just like we did in mitosis. Since we divide the cell in two different rounds, this ends up producing 4 cells at the end, 4 haploid daughter cells. Mitosis was the form of cell division wherein we started with a diploid parent cell and then we ended with 2 diploid daughter cells, and both of those daughter cells were identical. They were identical to each other and they were identical to the original parent cell. In meiosis, however, we start with the diploid parent cell and then we end with 4 haploid daughter cells, each of which is genetically unique, has a totally new combination of genes, and it's not like the other daughter cells that are created, and it's not like the parents. Obviously, they are genetically related. They will share some things in common, but each one has its own unique combination of these genes, and we'll see how in just a moment. The steps of meiosis. One thing you'll notice is that these words look familiar. Prophase, metaphase, anaphase. However, we've added a little Roman numeral to the end of each one. That's how you know that we're in meiosis. If I ask you a question about prophase, I'm asking about mitosis. But if I ask you a question about prophase 1, I'm talking about the first round of meiosis. So pay attention to those Roman numerals. Meiosis occurs in the same kinds of steps. Prophase 1, metaphase 1, Anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2. So that order of phases, P, M, A, T, it still holds here. We're just going to do it twice. However, there are some interesting differences between meiosis and mitosis that you're going to want to know. Everything unique about meiosis, everything that's special and interesting about it, occurs in the first round of division. It occurs during meiosis 1. Here are the steps of meiosis. In prophase 1, the centrosomes are going to migrate to opposite poles of the cell, and they're going to create spindle fibers behind them as they go. We could now call them meiotic spindle fibers, but they're spindle fibers made out of microtubule proteins, just the same as in mitosis. The nuclear membrane is going to break down, just the same as in mitosis. And the chromosomes are going to condense, and we'll actually be able to see individual chromosomes. So that's the same. So where's the difference? Here's the first difference right here in prophase 1. The homologous chromosomes start pairing up with each other. Okay, they pair up next to each other. Here's mom's chromosome 1, and here's dad's chromosome 1. Each one has 2 sister chromatids, and the chromatid, whatever's written on this chromatid here, if this says bald, then this one says bald also. Okay, so sister chromatids are identical, but we have 2 sets of chromosomes, homologous chromosomes. So dad's chromosome 1 lines up next to mom's chromosome 1. And here's the weird thing. They tangle up next to each other in a kind of romantic way. Mom's chromosome 1 and dad's chromosome 1 are going to tangle up with each other, but just on one chromatid. This is referred to as crossing over or synapsis. Synapsis is the fun way to say it, but I think most people are more comfortable with the idea of crossing over. What are they doing when they do this? What they're actually accomplishing is they're exchanging segments of DNA. So one of the little segments of mom's chromosome number 1 is going to get onto dad's chromosome number 1, and vice versa. So we end up with now 4 chromatids total, but each one is different. This one is all yellow. This one is all dad's, right here. This one is mostly yellow with a little bit of red, mostly dad with a little bit of mom. This one is mostly mom with a little bit of dad, and then this one is all mom. We have 4 unique chromatids at this point. So crossing over is the first difference between mitosis and meiosis. It happens in prophase 1. Then we get to metaphase 1. In metaphase 1, the homologous chromosomes line up next to each other in the center of the cell. Now, in mitosis, they lined up in a single file line, but here it's a 2-file line, 2 columns here. One homologous chromosome, then the next homologous chromosome, because the purpose of meiosis 1 is to separate these homologous pairs from each other, to cut the total number of chromosomes in half. So we're not going to break chromatids right now. We're not going to snap them yet. We are going to just separate out these homologous pairs. Then in anaphase 1, they are going to go to opposite cells. They're going to go to opposite poles and end up in different daughter cells. Now, when they line up in metaphase 1, we could have mom's chromosome 1 on the right and dad's chromosome 1 on the left. But it's just as statistically likely for them to go the other way around. And that's true for each pair. There's actually a really high number of different possible chromosome combinations that could end up going to each daughter cell at the end. Maybe one daughter cell gets 3 of one parent and 20 of the other one, or maybe it's more like 8 and 15. There's many different possible combinations of chromosomes that could end up in these 2 daughter cells after anaphase 1. So in anaphase 1, we separate the homologous pairs. Okay, that's different than in mitosis. This is an image of crossing over in action, so you can see the 2 sister chromatids lined up right next to each other, and then these points of crossing over, where it crosses over, we call them chiasmata, but chi is in the Greek letter X, so they're crossing. So it's chiasma, so it's crossing over. And we're just going to exchange segments of DNA. Now, because homologous chromosomes have the exact same types of genes in the same locations, whatever you swap from one to the other, it's going to be an equal exchange. So both chromosomes at the end are going to end up with a full set of genetic information. They'll just have a couple of each other's genes swapped around there. Review. Prophase 1, homologous chromosomes pair up, which we call crossing over or synapsis, and they exchange bits of DNA. In metaphase 1, homologous chromosomes line up side by side, and each one is going to face a different pole, and that's random assortment, independent assortment. We are going to separate these homologous pairs from each other, and each daughter cell is going to get a random arrangement of parental chromosomes. Then in anaphase 1, this is our reduction division. We've separated the homologous pair, so each daughter cell is going to end up with just 23 chromosomes. Hence, they are haploid. The daughter cells are haploid after meiosis 1. Now, Meiosis 2 is actually very simple. It's straightforward. It is exactly like mitosis. The main difference between Meiosis 2 and mitosis is that we start with half as many chromosomes. We start with haploid cells and we end with haploid cells. So, at the beginning of Meiosis 2, prophase 2, same thing happens as in mitosis. Nuclear membrane breaks down, chromatin condenses so we can see individual chromosomes, but you can actually see that these are chromosomes that have undergone crossing over because they have little different colored ends on the ends of them. And then the meiotic spindle is going to form. Spindle fibers are going to develop. In Metaphase 2, they line up on the metaphase plate right down the center of the cell. Remember, the metaphase plate is not a physical object. It's just the prime meridian of the cell. It's an imaginary line where everything is going to line up. In Anaphase 2, we break the sister chromatids away from each other. So up until this point, the chromatids have remained intact, but now we break the centromere and we separate the sister chromatids for each other. So each daughter cell is going to get just one chromatid at the end here. But since there were 23 of these two chromatid chromosomes, there will be 23 of these single chromatid chromosomes in each daughter cell at the very end. So in telophase 2 and cytokinesis, we form 4 haploid daughter cells, and each one is genetically unique. Because of crossing over, each chromatid is going to be unique from all the other chromatids. Because of random assortment, each daughter cell is going to have a different combination of chromosomes. Each one of these daughter cells is totally unique. These daughter cells then develop into gametes. They develop into either sperm cells or egg cells, depending on what kind of individual we are talking about. So here's my animation of meiosis. In prophase 1, we have crossing over. That's the big important thing. But we have the nuclear membrane breaking down. We have the chromatin condensing. We have the centrosomes migrating and the spindle fibers developing. In metaphase 1, the homologous pairs line up next to each other in a random orientation. Then in Anaphase 1, they divide, they go to opposite poles, creating 2 haploid daughter nuclei. We haven't broken chromatids yet, but we separated the homologous pairs. In telophase 1, we form 2 haploid daughter cells. And then we begin Meiosis 2. In Meiosis 2, the nuclear membrane breaks down. The centrosomes are going to migrate to opposite sides of the cell. The meiotic spindle is going to form, spindle fibers are going to develop, and they're going to pull those chromosomes to the metaphase plate. In metaphase 2, we have them lining up in a single file line, and then in anaphase 2, we separate the sister chromatids from each other. And after the sister chromatids separate, then we can form daughter nuclei, and in telophase, we pinch them apart into 4 haploid daughter cells at the end. Now, we started with diploid cells. 2n equals 8 in this case. Notice that here 2n equals 8. And then when we got to the end, we had haploid cells, 4 chromosomes each. N equals 4. So n is the number of chromosome pairs that you have. And in this case, we have 4 pairs. So n equals 4. Currently, we have 2n chromosomes, 2 of each pair, so 8 in there. And now, right here at the end of meiosis 1 is when we form haploid daughter cells, which only have 4 chromosomes each. So make sure you know the steps and in what order they come before you get to any kind of test here.